Hello everyone, this is your educator Ria from Biotechnica. So today I will be talking about Brassinosteroid Phytohormone, where you have to remember all the points that are listed in this mind map. So let's understand each and every point one by one. So coming to the introduction of this Brassinosteroid Phytohormone, so this phytohormone belongs to the family of sterol, which is lipid soluble in nature. Because you all know that sterol is a structure which contains many benzene ring, or you can say cyclic ring, that is soluble in lipid solvent, and hence brassinosteroid is lipid soluble in nature. So they were for the first time identified as a growth promoting substances in the pollen of Brassica napus, where they were discovered a compound called as brassin. That's why it is called as brassinosteroid, because it was the very first compound that was isolated or discovered in the pollen of Brassica napus, that is a derivative of sterol family and hence named brassinosteroid. Now, what's the most bioactive form of brassin in eukaryotes? It's brassinolide. Again, an important question for exam point of view, where you may be asked about the most bioactive form of brassinosteroid. It's brassinolide, which is similar to the animal hormone that is called as polyhydroxylated steroid. So, shall we say this brassinosteroid is a polyhydroxylated steroid as it contains many hydroxyl group in its steroid ring. Now, you all know that the optimal concentration of hormone is important for the proper growth and development of the plant. So, what happens if the concentration of regulator exceeds the homeostatic control? Now, here happens the regulation of this phytohormone. So, there are three regulatory mechanisms that are well known to cause to maintain the optimal concentration of this phytohormone inside the plant body. So what are these three mechanisms? The first is inactivation, where brassinosteroid is inactivated by some means. The second mechanism is catabolic reactions, which include epimerization, oxidation, hydroxylation as well as sulfonation. So what happens in epimerization? So epimerization is a process in which there will be a carbon configuration around a single carbon in a structure of brassinosteroid. What is oxidation? Oxidation is the addition of oxygen molecule. Hydroxylation is the addition of hydroxyl group, that is OH negative group, whereas sulfonation is the addition of sulfonic acid to the structure of brassinosteroid. Now, what's the formula of sulfonic acid? It is SO3H. And the third reaction being conjugation, where this brassinosteroid is conjugated to hexose sugar like glucose or some kind of lipid, which results in the inactivation of brassinosteroid, and that's how this brassinosteroid is maintained at the optimal level in the plant cell. Now let's understand the biosynthetic mechanism of brassinosteroid. Now, what's the precursor for the synthesis of brassinosteroid? So the name of the precursor is a sterol compound, that is campestrol. So you have to remember the name of the precursor molecule, that is campestrol, which is very similar to the animal hormone, that is cholesterol. That means campestrol in plants is equal to cholesterol in animals. So both are derivatives of sterol compound. Now what's the enzyme that is needed for the biosynthetic mechanism? It's cytochrome P450 monooxygenase, which is also known as ER enzyme, that is endoplasmic reticulum enzyme. That is why the location of this biosynthesis is endoplasmic reticulum, because enzyme is present in this organelle itself. So what happens? The pathway starts with the precursor, that is campestrol. So firstly, this campestrol undergo a series of reaction so as to form campestanol, 6-deoxycathesterone, 6-deoxytestosterone, and finally it results into the formation of active brassinosteroid, that is castasterone. Now this is the active brassinosteroid, which undergo again one reaction so as to form the most bioactive form of brassinosteroid. As we have already seen, this is brassinolide. Now suppose if you get a question in exam like which is the most bioactive form of brassinosteroid, and now you have four options in which two options are brassinolide as well as castasterone. So you have to select brassinolide because it is the most bioactive form of brassinosteroid, and this castasterone, it is also the active brassinosteroid, but you have to select this first. Now coming to the functions of this brassinosteroid. So this brassinosteroid serves many different functions in the plant cell. The first function is developmental process, as it involves in cell division, cell elongation, cell differentiation, as regulating the functional activity of the plant cell. It suppresses the photomorphogenesis mechanism. It is also involved in the reproductive development, helps in the germination of seed, also promotes the leaf senescence, that means the aging process of the plant cells, and is also involved in the stress response. So whenever plant is facing any kind of biotic and abiotic stress, this Brassinosteroid phytohormone is regulating the stress response in the plants. It also known to inhibit the germination process. It is also known to reduce the ability of the ABA to inhibit the germination. Now we all know this. There is one phytohormone that is what called as ABA, which is also known as abscisic acid. This is used to inhibit the germination of seeds in plants. This inhibits the germination of seeds. So what happens once this brassinosteroid is synthesized in plant? It shows its inhibitory effect on this abscisic acid. That means there will be no delaying of germination, and germination of seeds will happen in plants. So it has a negative effect on the abscisic acid phytohormone, where it was known to inhibit the germination of seeds. It is also known to inhibit the root hair formation. Now we all know that auxin phytohormone is responsible for the growth of root as well as the root hair. So what happens, how this auxin signaling is on inside the plant? So in every signaling, there is one inhibitor that is keeping the signaling off. So how this signaling is on? Once this inhibitor is cleaved by some other means. So if this inhibitor is cleaved, this signaling will be on. So how the auxin signaling is on? If the inhibitor called as OX/IAA is cleaved in auxin signaling. So once the inhibitor is cleaved, the auxin signaling is on. That will promote the growth of roots as well as root hair. So what happens in case of brassinosteroid signaling? Once the signaling is on, that means the brassinosteroid is synthesized in the plant cell. So what it will do? It will increase the synthesis of this inhibitor, which is called as OX/IAA, which is one of the inhibitor of auxin signaling pathway. So what it will happen? It will shut down the auxin signaling and there will be no root growth formation. That's why this phytohormone is known to inhibit the root hair growth, because it is indirectly increasing the production of an inhibitor, that is OX/IAA, which is involved in auxin signaling pathway. So it also helps in the pollen tube germination, which is one of the most important function for which brassinosteroid is known. It helps in the pollen tube germination. It also promotes the development of female gametophyte, as well as it accelerates senescence, that means aging process of the plants. So this is all about the functional profile of this phytohormone. So last we'll understand the signaling mechanism, how this brassinosteroid induced response is shown in the plant cell. So this is how we have to study the signaling mechanism of this phytohormone. So let's understand that. Now, what's the name of the ligand in this signaling pathway? It's brassinosteroid, that is BR or BL. What's the name of the receptor? Is BRI1, which is also called as brassinosteroid insensitive 1. Insensitive 1. What's the name of the inhibitor? It's BKI1, which is also known as BRI1 kinase inhibitor 1. So this is the inhibitor for this signaling pathway, which is called as BKI1. What is the name of the phosphatase? That is BSU1. And what's the name of the kinase? That is BAK1 as well as BSK, which is also known as brassinosteroid signaling kinase. So this is the main kinase from where the signaling starts in plants. Brassinosteroid signaling kinase. So here you have to remember the short forms or the short names of all these inhibitor, phosphatase, whether it is kinase, ligand as well as receptor. So now let's understand the signaling mechanism. So what happens? Suppose this is a plasma membrane. This plasma membrane has a receptor that is what called as BRI1. These receptors are in the form of inactive monomer. So they are in the form of inactive monomer. Along with it, there is one receptor kinase that is embedded in the plasma membrane, that is what called as BAC1. So again, there will be a receptor kinase that is called as BAC1. So this is again in the form of inactive monomer. So now the signaling is off. Why the signaling is off? Because there is no bracinosteroid in the plant. So why this signaling is off? Because there is one inhibitor that is linked to the cytoplasmic tail of this receptor. And what is the name of the receptor? It is BRI1. That is Brassinosteroid insensitive 1. And what's the name of this inhibitor? It is BKI1. So this BKI1, which is an inhibitor, is associated with the cytoplasmic tail of the receptor that is responsible to initiate the signaling pathway of this mechanism. So what happens once the bracinosteroid binds to this? So suppose bracinosteroid is binding to this receptor as well as receptor kinase. So here happens the activation of the signaling pathway. So while the signaling pathway is activated, this kinase enzyme is activated. Now what's the function of this kinase? The function of this kinase is to add the phosphate group to the receptor. Now this kinase enzyme will add the phosphate group to its target. Now this is the target for the kinase enzyme. Now the phosphate group is added. Once the phosphate group is added to the receptor, it is now activated. Now the receptor is activated and the signaling mechanism will be on. Once the signaling mechanism is on, this BKI1 is removed with the help of some other protein that is what called as 14-3-3S type of protein. How this protein is activated? Because the receptor is phosphorylated by its target, that is BAK1 kinase. Now the signaling is activated and the inhibitor is relieved from the receptor, that is BRI1. Now this BAK1 will activate another sort of kinase which is called as BSK. Now this will activate BSK. So once the receptor is fully activated, now it's the time for the activation of signaling kinase. So this is called as Bracinosteroid signaling kinase, which will be activated, which will activate some other target called as BSU. Now this BSU is the suppressor of the bracinosteroid, which is activated with the help of BSK1, and it will phosphorylate it. Once this is phosphorylate, it will act on some other protein, that is BIN2. Now here, in a normal form, this BIN2 exists in its phosphorylated form and it's active. Now once BSU is activated, this will act on BIN2 protein and will cause the degradation of this BIN2. So this BIN2 is now dephosphorylated because this BSU is a phosphatase. This phosphatase is activated when the phosphoryl group is added. Now once this is activated, it will act on its target, that is BIN2. अभी BIN2 का क्या होगा? Dephosphorylation. Now this BIN2 is dephosphorylated and will undergo 26S proteasomal degradation. Proteasomal degradation means this pathway is now free from BIN2. Why it is called as now free from BIN2? Because when the signaling was off, this BIN2 was suppressing two different transcription factors, that is BZR1 as well as BES1. So it was suppressing two transcription factors and the signaling was off. But if BIN2 is not there, it is cleaved by suppressor of phosphatase. Different type of transcription factor, that is BES1 as well as BSR1. So both these transcription factors are inactivated. Now this BIN2 is cleaved by this suppressor because phosphate group is removed, means this is deactivated. Now what happens? These both transcription factors are free. It is free from any BIN2 protein. So they will translocate inside the nucleus. So these BES1 as well as BZR1, they will translocate inside the nucleus and will bind to the response element of the DNA, where they will initiate the transcription of the genes, that is brassinosteroid related gene expression. And this is how brassinosteroid induced function is shown in the plants. So I will repeat this phenomena or signaling cascade. So what happens when the signaling is off? So brassinosteroid receptor, that is BRI1, whereas the receptor kinase, that is BAK1, they are there in the membrane in the form of inactive monomers, and the signaling is off. Why the signaling is off? Because there is one inhibitor that is binded to the cytoplasmic tail of the receptor, that is BRI1. And what's the name of the inhibitor? It is BKAI1, that is inhibitor kinase. So it is binding to the cytoplasmic tail of the receptor, thereby shutting down this signaling mechanism. Now, once the brassinosteroid binds to this receptor kinase as well as BRI1, so what happens? This will activate one kind of protein, that is what called as 1433S, that will relieve this receptor from any kind of inhibitor, that is BKI1. Now this BAK kinase, it will be activated because that was present on the membrane. Now this kinase will be activated, that will cause the phosphorylation of its target, that is BRI1. Now this receptor is phosphorylated, which results into the dimerization of both the receptor as well as the BAK1. Now both the inactive monomers, they will dimerize into each other, that will form heterodimer. Why heterodimer? Because two different units are binded to each other. One is BRI1 and the other is the receptor kinase, that is BAK1. Now this pathway is fully activated because receptor is free from any kind of inhibitor. Now this BAK1, which is a kinase, it will activate another signaling kinase that will actually starts the signaling process. Now what's the name of that kinase? It's BSK. Now once this kinase is activated, it will activate its target, that is BSU. And here you can see BSU ka naam kya hai? BRI1 suppressor. That was suppressing the signaling mechanism. Now this will be phosphorylated with the help of the kinase, that is BSK. Once this is phosphorylated, it will act on its target, that is BIN2. Now BIN2 in its normal form, when the signaling was off, it was in its phosphorylated form, which was suppressing two transcription factor, that is BES1 and BZR1, so that it do not translocate inside the nucleus and doesn't activate any kind of signaling process. But now this BIN2 is a target of this phosphatase, that is BSU. Now what's the function of phosphatase? It will always remove phosphate group from the target, and the target here is BIN2. And now from BIN2 this phosphate group is released, and means BIN2 is inactivated. Once this is inactivated, it is marked for 26 as proteasomal degradation, where it is cleaved into smaller peptides. And now the transcription factors are free, that is BES1 as well as BZR1. Now they will translocate inside the nucleus, where they will bind to the DNA segment. कहाँ पे binding होगा? Response element. Suppose this is a DNA segment, it will bind to the response element and will induce the brassinosteroid induced gene expression. And this is how the brassinosteroid signaling is happened in the plants. So this is all about the signaling pathway. So what's the type of the pathway that is happening in case of brassinosteroid? It is serine threonine kinase signaling, where happens phosphorylation of kinases as well as dephosphorylation of many target like BIN2 protein. And phosphorylation किसका हो रहा है? BAK का हो रहा है, BSK as well as BSU, which is a phosphatase for the target, that is BIN2. So this is all about the signaling mechanism. And we have completed with phytohormone brassinosteroid, where we have seen the introduction, the discovery, what's the nature of this phytohormone, what's the active form, how it is regulated in plant, as well as the functional profile of this. And along with this, we have also seen the biosynthetic mechanism, where we have seen the campastrol, it it is acting as a precursor for the synthesis of brassinosteroid. So thank you everyone for watching this video. If you like the video, do not forget to like, share and subscribe to the channel, that is Biotechnica. Meet you in the next video with different phytohormone. Till then, take care. Bye-bye. Have a nice day. Keep learning.